We're moving into one of my favorite parts of genetics, and I hope you all agree, but I'll try to convince as many of you as possible that translation is one of the most interesting parts of genetics, because that's where we're going to see how messenger RNA becomes or encodes proteins, the things that actually do the job of the cell. And before we get there, we want to finish up with transcription, processing in particular, and make sure that if anyone has any questions about vocabulary or tasks so far, that you've got a chance to do that. Right now, we've talked about the primary transcript and messenger RNA. Messenger RNA is what exists after processing. And the rest of these vocabulary items, hopefully we'll get through today. We'll see. We did talk about introns, exons, and different forms of splicing. So those are all related to splicing down there. We did that last class. Today we're going to move on to the other forms of RNA processing and maybe start translation a little bit. We worked last time on finding splice sites. A 5' splice site, GU, with a branch point somewhere in the middle, and an AG is the 3' splice site. So that was the first task. We're going to move on to identifying possible outcomes of alternative splicing. We talked about that a little bit last time as well. So, any questions? Okay. So last class we ended with this sort of an activity. We had a theoretical 3-exon, 2-intron transcript. This is not a messenger RNA yet. This is a primary transcript. We'll reserve the word messenger RNA for a processed transcript. So in eukaryotes, that means splicing has occurred, plus what we're going to talk about today is the addition of a 7-methylguanosine cap on the 5' end, and then the addition of a poly A tail on the 3' end of the transcript. And at that point, and only at that point, with the addition of, say, splicing, the addition of the cap, and polyadenylation, the addition of the 3' tail, would we refer to that as a processed messenger RNA. So I want to show you a few examples of student responses from class last time, possible ways to splice that 3-exon, 2-intron transcript. So there's these first 3 slides, I'd like to get your feedback. Are these accurate? Are these possible splice variants that we can get from that initial gene? Love to get feedback. All anonymous. So this one's okay? Is this all of the possibilities for splicing that 3-exon, 2-intron transcript? So it could not be spliced, right? None of the introns could be removed. That is the mature messenger RNA after capping and polyadenylation. You can remove the first exon. You could remove the second exon, but not the first. You could splice out all the introns, and you could do the skipped splicing or the intron excision, where you get rid of exon 2 along with both of the introns. Are there any other possible ways to splice this transcript? What about these? And let's assume that this means that splicing did occur and the introns had been removed. So if that was the case, are these different splice forms of that transcript? What's happened with some of these splice forms? The exon order has been rearranged so that, for example, on the bottom we've got exon 3 coming before exon 2, coming before exon 1. So again, I'm assuming that this is still in the 5' to 3' orientation, but now the exons are being moved around in different orders. Is that something that happens in the cell? Not usually. Exons do tend, even though all of these alternative splicing things can occur, exon skipping, intron retention, the order of exons tends to stay in the left to right 5' to 3' order. Not always. There's always exceptions, and you can, if you want to, go find some literature that explains some rare instances where people found exons spliced together out of order. So in general, in other words, and we can definitely debate this. So if you are the student that drew this, you're not wrong. It's just not common for this to happen. In general, that we would expect. You wouldn't expect to see exons 2 and 3 switch place, or 1 and 3, and so forth. So that top one, yeah. The bottom ones maybe could happen, but really rare, and we just, for the sake of this class, won't consider that those types of things can happen. Any questions about that? Yeah?
I know you're saying not considering 3, 2, 1, but what would it look like for that to happen?
Right. So if this sort of thing were to happen, you would probably have to imagine some sort of a scenario. The DNA loops. I don't even know if I can do this in a reasonable way, but if that's the 5' end and the 3' end, you might get 2 splice sites that are in different exons close to each other. I don't know, maybe like that. That would rearrange, because DNA is a flexible object, it's like cooked spaghetti, so it can loop around, and you might accidentally inside the nucleus get a 5' splice site and a 3' splice site from 2 totally different parts of the gene, parts of the transcript near each other, and they might splice in an odd conformation. That's a hand-waving argument. I'm telling stories now. That's one way that it might happen. And that's not even a very accurate diagram. It's just trying to represent what I'm thinking in my mind. But I'm not a very good artist either. Okay, what about this case? This is why I asked for student feedback, because I like the diversity of thoughts. I challenged you to be creative about how to do this, and I'm glad to see this creativity. Well, yeah, first of all, it's exon, not extron, that's fine. Just minor terminology, that's fine. Exon 1, intron 1, can this happen? Can you get rid of a terminal exon by splicing? That is, initially it had looked like this, and splicing happens like that. Gets rid of the first exon and the first intron. What would have to be true for, we know about splice sites, we know about 5' and 3' splice sites. So what would have to happen for this to be a 5' splice site?
The exon would have to begin with GU.
There'd have to be a GU somewhere there to be a 5' splice site to splice together with that 3' splice site. And that's why the first exon, although, again, this is not a rule, this is common, first exons don't tend to get skipped because there's nothing to the left 5' of that first exon to act as a splice donor, the 5' splice site. There's no DNA there, that's the start of the transcript. But there could be, and this does sometimes happen, there could be a, what we call a cryptic splice site, a GU that is actually in an exon that can occasionally get used for splicing. And so maybe rarely this does happen, that the first exon gets removed as part of splicing. Pretty rare, but maybe rarely could happen. So any questions about any of these sorts of splice forms, splice variants? So typically introns get removed, sometimes not all of the introns, sometimes just a handful of them. Sometimes introns get moved in addition to exons. And what I finished up with, I just wanted to show a brief data-based version of this argument that I left you with at the end of last class. What is the benefit of splicing again?
Diversity.
To create diversity in transcripts. And this is just proof of that. This is from an old review paper from 2002, but so even 2 decades ago, this was still true. If you looked in humans, for example, here on the left, this first column indicates what? The percent of messenger RNAs with AS alternatively spliced forms. So almost half of our 30,000 genes that are produced from our chromosomes undergo this sort of processing. So it's not like alternative splicing is a rare thing. Almost half of our genes undergo this sort of process. So you might find that different cell types turn on the same gene, but there are cell type specific splicing patterns. So that maybe in your eye, intron 1 gets left in, but in your toe, intron 2 of the same gene gets left in. It's weird, but it happens, and it happens a lot. And so you can see that this varies depending on what sort of organism you're looking at. Alternative splicing, not so prevalent in a lot of species, but is in some others. Do prokaryotes, bacteria basically, do RNA splicing or capping or polyadenylation? No. So this is just, answer here is eukaryotes. The eukaryotes do RNA processing. There are some forms of RNA tinkering that bacteria can do, but it's not capping, splicing, polyadenylation. Well, at least not splicing. Okay. So what about, this one is a bit misleadingly worded. What are the 3 steps? So I should have underlined maybe processing when I asked this on Monday. 3 steps involved in processing. What are they? Processing. Capping, splicing, polyadenylation, the addition of the 3' tail. So that would be B, D, and F. What are A, C, and G? Those look like they got a lot of votes too.
Those are
Initiation, elongation, and termination.
transcription.
Yeah, those are the steps in transcription. That's just not processing. That's the first step. You do initiation of transcription. RNA polymerase starts making the transcript. Elongation, RNA polymerase makes the transcript. And then termination, RNA polymerase stops producing transcript. And then, that's the first step. A, C, and G. Initiation, elongation, and termination. Splicing happens in eukaryotes at the same time transcription is happening. But in here, it's capping, polyadenylation, and splicing are the process. Benefit of adding the 7-methylguanosine cap, and I'll talk about the details of what that means in just a minute. So you're absolutely right, at least most of you were. It's D. Prevents messenger RNA degradation. This is one chemical modification to transcripts that cells use to prevent the messenger RNAs from being destroyed quickly by the cell. There's another right answer up here. Improve translation. We're going to see why the cap is important for translation today or Monday. What about splicing? Generates diversity. Option B. Does it make the RNA smaller?
Yes.
Does removing introns make a messenger RNA smaller? Yeah. But is that a benefit? Right. We could debate whether it's a benefit, but that's why 5A isn't my choice of answer for this. It's true, but whether or not it's a benefit is up in the air, unclear. And then the polyA tail definitely, looks like most of you got that, prevents degradation, the benefit of having that tail. So what do I mean by capping, splicing, and polyadenylation? We've already talked splicing. So what about the 7-methylguanosine cap? So quick review from the videos. If you have RNA polymerase transcribing a messenger RNA, let's say that's in blue, as soon as that transcript, RNA, starts being produced, it's got a 5' end. Turns out there are enzymes in our cells that are basically purposed to destroy RNA molecules. Why would we want to do that? Say it again. Viruses. So there are RNA viruses that can infect our cells. So some of our cells have proteins that look for and destroy RNA, because a lot of viral genomes are RNA genomes, and we don't want them invading our cells, so we have some machinery that destroys RNA. Except we don't want the messenger RNAs that we're producing to be destroyed at the same time. So that involves, in this case, the addition of this weird linkage where two nucleotides are joined at their 5' carbons. So normally, during synthesis, you have the 5' end of one nucleotide. Let's say it's an A. I know that's not what an A looks like. It's got a 3' end. It'll be joined to the 5' end of the next nucleotide. Say it's a G. Right? So nucleotides have 5', 3' linkages. This is the only exception, is that there's a specific enzyme, which you don't need to know the name of, that adds a version of G, which is methylated on carbon 7. 7-methylguanosine gets added, its 5' end links to the 5' carbon of the first nucleotide in the RNA. The benefit of this is that now the 3' end of that 7-methylguanosine is hanging out. It's the thing that's being presented to the cell, 3' end. That prevents 5' endonucleases, enzymes that grab onto 3' ends of RNAs can chew in from the right side. If we have a 5' end on the RNA, there are specific enzymes that will grab onto 5' ends and start chewing up, degrading, removing nucleotides from the 5' ends of RNAs. This prevents that 5' end of the new RNA molecule from being recognized as RNA. There's no 5' end anymore, there's just another 3' end. And there are no enzymes, well, there are no foreign enzymes that can cleave that sort of bond, the 5' linkage. All you need to know. So it protects the RNA from being degraded. There are a couple other things this does too. Does anybody know the cap is used for other than protecting the RNA? It's important in a couple other processes in eukaryotes. Where does transcription happen in eukaryotes? In the nucleus or in the cytoplasm? Where's the DNA? It's in the nucleus. So the RNA polymerase is in the nucleus reading the DNA. Where does translation occur? In eukaryotes. The reading of the messenger RNAs and producing proteins occurs in the cytoplasm. So the messenger RNA starts in the nucleus and it has to get out into the cytoplasm to be translated. That means that there are proteins in the nuclear envelope in the membrane that surrounds the nucleus. There are proteins there that grab messenger RNAs and actively transport them into the cytoplasm. And those transport proteins recognize the 7-methylguanosine cap as targets for pulling messenger RNAs out of the nucleus and putting them into the cytoplasm for translation. And then after that, so that's one additional value of the 7-methylguanosine cap, and after that, translation in eukaryotes relies on, we'll see this next class, relies on that 7-methylguanosine cap to start translation. So they have many uses. So what I'd like you to take away from this is you just have to know that a processed eukaryotic transcript will have this 5' cap, 7-mg, 7-methylguanosine. If it doesn't have it, it's not a protein coding transcript. It's an RNA molecule that does something else, maybe like a ribosomal RNA. And then as RNA polymerase finishes up translation, there's that RNA molecule. At this point, it's got its 7-methylguanosine cap on it, still being produced by RNA polymerase. So RNA polymerase is moving that direction. Got 2 strands of DNA. The last step in processing, after capping and splicing, is the addition of that 3' tail. So what's going to wind up happening is we're going to have a mature transcript with the cap and a bunch of A's on its 3' end. Those A's are there to keep these enzymes, which I guess I'll diagram as sort of Pac-Man-like things, from grabbing on. These enzymes do exist. They grab onto the 3' ends of RNA molecules and they start removing nucleotides. They start chewing up the RNA molecules from their 3' ends. So presumably the presence of this poly A tail is to basically have some nonsense nucleotides that these enzymes can act on that don't do anything to the transcript until that enzyme winds up having eaten up all those A's and finally starts chewing on the actual coding region of the transcript. So the addition of the poly A tail essentially delays degradation of messenger RNA molecules. It means they can stick around in the cytoplasm longer and be translated more. And one thing I'd really like you to remember, this is critical for genetics, is this is really important. This is how the cell knows when to stop transcribing. How did RNA polymerase know where to start transcribing?
The promoter.
So something binds to the promoter, TBP binds to the TATAAA sequence, brings RNA polymerase. That's where RNA polymerase knows where to start transcribing. How does it know where to stop? This is really important to know. There's going to be, what sort of a signal will tell this protein, RNA polymerase, where to stop transcribing? Is it going to be another protein? Is it going to be a hormone? What sort of a signal can RNA polymerase use to know when to stop making more RNA? Pardon? It's going to be DNA. There's a specific DNA sequence that RNA polymerase recognizes, and its related proteins that are associated with it detect, and that tells RNA polymerase where to stop. So that signal sequence is the polyadenylation signal sequence. So if the top strand is the strand that RNA polymerase is reading, that's the template strand, which means the bottom strand of DNA there is the coding strand. So what's the sequence present in the RNA molecule that's related to the poly A signal sequence in RNA letters, RNA nucleotides? So that's the question. Is it the same as the template strand, or is it the complement of the template strand? The coding strand is the one that reads the same as the RNA transcript. So in this case, that sequence would be complementary to the template strand. AA, which? U. AAA. That is what you look for in the RNA molecule that is the polyadenylation signal sequence. So it's present in the DNA too. This is a sequence that's in the gene, the DNA, but you'll also see it at the end, near the end of a messenger RNA molecule. So there are proteins, these circles or blobs that I've associated here with RNA polymerase. One of them is responsible for recognizing the polyadenylation signal sequence. And the next one acts as sort of a molecular scissors that cuts the RNA a few nucleotides, maybe 15 to 20 nucleotides downstream. Let's say 15 for the sake of this. 15 nucleotides, 3' of the polyadenylation signal sequence. In other words, in this messenger RNA I drew on the bottom in blue, the final version, there would be an AAU, AAA, and then there would be about 15 to 20 nucleotides before you would see the start of this poly A tail, just a bunch of A's connected together. In other words, this AAU, AAA was the signal that RNA polymerase would stop transcribing. It had transcribed about 20 nucleotides before this protein up here that recognizes that sequence spots it and causes the RNA to be cut a few nucleotides downstream of that sequence. Then the enzyme poly-A polymerase comes along and adds those A's to the 3' end. So poly-A polymerase, many A's, poly-A, that's its job. It's a DNA polymerase or an RNA polymerase that just adds a bunch of A's. A. Doesn't use a template. It just sticks A's onto things.
Dr.
Yeah.
Are there any like similarities in origin between like the poly-A tail and telomeres? Since they do similar.
Right, so that's interesting. Compare and contrast. Poly-A tails versus telomeres. Telomeres are the extra bits of DNA at the ends of chromosomes that prevent degradation at the ends of our chromosomes. So it's a good comparison. Contrast-wise, the difference between telomeres and this is that poly-A polymerase, this enzyme that adds the A's, doesn't need a template. It doesn't read, in other words, there's no poly-T that it copies to add the A's. But that's true in telomeres. In telomeres, there is a template that telomerase uses to add the telomere sequences. But otherwise, very similar. Good question. Any other questions? So at this point, we've seen the three different versions or steps in messenger RNA processing: addition of the cap, addition of the tail, and splicing. And we've also seen a lot of RNA and DNA elements that are important in how the gene regulates what proteins do things when during the process of transcription and translation. Now that was eukaryotic termination. Prokaryotes, we haven't really focused on in class. I just wanted to mention this for completeness because if you go on to one of Dr. Yoon's classes, or if you're already taking one of, say, Dr. Yoon's classes, you might want to know something about prokaryotic transcription termination as well. There are two different versions of it, but here's the main one. One of the main ones is that when RNA polymerase is transcribing an RNA molecule, there are sequences in the ends of bacterial genes that form secondary structures called hairpins. So this is the RNA molecule. And what that would look like if I blew it up down here on the bottom of the screen is a series of nucleotides that base pair with each other, because remember, RNAs are single-stranded. They don't have anything to base pair with. So their hydrogen bonding atoms are just sort of looking for things to base pair with. And if an RNA molecule has a sequence like, I'll just make one up, ATC, and there's a GAT somewhere nearby in the sequence of that same messenger RNA transcript, then they'll base pair with each other and form this stem loop or hairpin shape. And it turns out that in transcription, what happens is that that shape physically interacts with RNA polymerase and causes RNA polymerase basically to fall off of the DNA. This is kind of like a lock and key mechanism. So the end of every prokaryotic gene, or most of them, for the ones that use this form of transcription termination, that physical structure, this shape, can actually interact with proteins that cause RNA polymerase to stop transcribing. So these ones don't have polyadenylation signal sequence. They don't do the polyadenylation. This is how transcription terminates in many prokaryotes. So there is a specific sequence, in other words, in eukaryotes, AAU, AAA. But in prokaryotes, the sequence itself isn't important. It's the structure of the sequence. So you could think of many other sets of nucleotides here that you could use to form the stem of a hairpin. Any complementary nucleotides that are in the right orientation would work. So there isn't a specific sequence to look for. There's a specific shape of RNA to look for if you were trying to identify transcription termination signals in prokaryotes. So I think that means we've covered all the vocabulary and tasks to this point. Please look over those by Monday. See if you have any questions, things that you don't feel I covered well or at all. But then we get to move on to translation. Again, my favorite topic for the central dogma, because this is where proteins get produced. The final outcome of DNA makes RNA makes protein. So we've got a bunch of vocabulary items. You've probably heard many of them before, and we just go, hopefully you'll find we go more in depth about translation and the effects of mutations on translation in genetics. You've probably seen this in biology 1. You probably saw this in high school, maybe. But there's a lot of ways that translation can go wrong, especially with genetic mutations, and that's kind of the focus of this topic. So how does translation start in eukaryotes? There's a really important protein that does translation. Do you know its name? Set of proteins, actually. It's a protein complex. Starts with R, ribosome. The ribosome is the set of proteins that does the job of translation. It's true in prokaryotes and eukaryotes. Ribosome. So the whole concept of this initiation of translation is how does the ribosome know where to start reading the RNA molecule? Does it start at the 5' end of the RNA and just start reading nucleotides and producing proteins? That's not the case in eukaryotes or prokaryotes. So quick compare and contrast. So let's say prokaryotic is on the top and eukaryotic's on the bottom. How does the ribosome know where to start reading the RNA molecule? In eukaryotes, there are two subunits. It's actually true in prokaryotes as well. Small and large subunits of the ribosome are. They grab onto, or at least one of the subunits does, grabs onto the 7-methylguanosine cap. That's why this cap is essential for translation. If an RNA molecule doesn't have that 7mg, the ribosome can't bind to that RNA molecule and try to translate it. It just won't happen. And that's why earlier in class we said that the cap not only protects the RNA from degradation, but it's also important in translation efficiency. If you don't have it, the RNA doesn't get translated. So then what happens to the ribosome? Sitting there on the 7-methylguanosine cap, ready to make some protein. How does it know where to start translating? Do you know what the first amino acid in every protein is? It's, what is it? Methionine. Okay, so that tells us, if you know that, then that tells us that the ribosome has to find whatever part of RNA encodes methionine. So we're going to talk about the codon table in a little bit, or maybe on Monday, definitely on Monday. The codon for methionine, the 3 nucleotides in RNA that encode methionine are AUG.
Like August.
Like August?
AUG.
Okay. Is that related to methionine? I mean, it is. It's the codon for methionine. So remember that.
Popped in the
Mnemonic.
Don't mind me.
No, that's totally fine. So August. How to make that a better mnemonic? The ribosome, let's try this. The ribosome falls down the RNA molecule, August, until it reaches the first methionine codon, the first AUG. The ribosome binds the cap, and then it reads the RNA molecule from 5' to 3' until it reaches the first AUG, and that is what is affectionately referred to as the start codon. That's the spot where RNA, or the ribosome starts producing proteins. So we'll start producing a protein with methionine at its, methionine is going to be at the 5' end of the protein.
No, that'll be the 3' end.
And I was wrong, you're wrong. No, it's not 5', 3'. What's the polarity notation for proteins?
Carbon and nitrogen.
Yeah, it's amino or N terminus. So the amino terminus of a protein is M. Pretty much guaranteed, maybe a few exceptions. So the first amino acid will be an N, M. Amino nitrogen, yes, but M encodes methionine. Let's make that a little bit less confusing. Okay. And then the ribosome keeps moving down the RNA in steps of 3 nucleotides and reading the nucleotides and producing proteins. We'll talk about the codon table in a minute. How does this change when you're dealing with prokaryotic RNA that doesn't have a cap? No 7-methylguanosine cap. The ribosome doesn't bind the 5' end of the prokaryotic RNA. It does something else. It looks, and I challenge you all to come up with a good mnemonic for this one. Not just a season, but something.
Does it look for the hairpin?
In this case, there's another, wait for it, consensus sequence. Oh, sorry, 1A. AGG, AGG, U. Which is, you may have heard this at some point in the past, the Shine-Dalgarno sequence. I'm assuming that that's the people or person that found it and not some other words in some other language, I don't know. Shine-Dalgarno sequence. So bacteria have this particular sequence. It's a consensus sequence. So again, like we've seen before, the ribosome maybe doesn't have to have that exact sequence. Maybe it has to be pretty close to that sequence for the ribosome to bind. This is a consensus sequence. So it might be AGG, TGGU, UGGU. That might be fine in some instances. So the ribosome will bind there, the Shine-Dalgarno sequence, and then start translating. So before we move on, please ask questions if you want. Think about this for a second. This means something really important to the process of transcription and translation. Is every part of the messenger RNA molecule translated?
No.
What part isn't translated? In eukaryotes, say. Verbal description. What part of this transcript, this messenger RNA molecule, isn't translated?
Everything that comes before the start codon.
Everything that comes before the start codon is not translated. And so we refer to that as, we'll see this many times, the untranslated region. See, sometimes geneticists come up with really horrible terms for things, and other times we make it way too simple. Untranslated region. I capitalized the T on purpose because we abbreviate this UTR. In this case, it's on the left side of the molecule, the 5' end, so this is the 5' untranslated region. Everything between the cap and the first start codon won't be translated. The ribosome scans right past it, doesn't turn it into protein, or doesn't read protein. It goes to the first AUG, and that's where translation starts. So everything to the left is untranslated.
Dr. Rowe,
Yeah.
is there something about like a 12 initiation factor for eukaryotes? Like, is it a 12-step initiation factor?
For translation,
Or is that something else?
I think that's something else. Initiation factors tend to be transcription initiators, but if you find something about that, let me know, because I will clarify later if need be. So that's translation initiation, prokaryotes versus eukaryotes. Shine-Dalgarno sequence versus 7-methylguanosine cap. Eukaryotes do ribosome scanning. It starts at the first AUG. Prokaryotes don't do that. The Shine-Dalgarno cap tends to set the ribosome at its AUG. So they still use AUG as a start codon, but the ribosome arrives at the start codon in different ways. So if that is a eukaryotic transcript, what protein does it produce? Here's the codon table, and we know that ribosomes read codons, groups of 3 nucleotides. 3 consecutive nucleotides, that is a group of 3 all in a row, 3 consecutive nucleotides. So if you were a eukaryotic ribosome, what protein does this messenger RNA encode? Okay, so the ribosome binds the 5' cap, scans until it finds the first AUG. So that's methionine. And then it moves how many nucleotides before it reads the next codon? This is the important part. You can either read AUG, and then UGC, and then GCG, and then CGA, or
Next sequence of 3.
it's every 3. So don't let what I just said throw you off. It reads every next group of 3 nucleotides. So the next set of 3, CGA, encodes, we look in the codon table, arginine, and then we would need more RNA molecule to keep going. So far we've got Mr. or methionine arginine. So the ribosome moves every 3 nucleotides reading codons, and then they get translated based on what we know is the codon table. Which sets of 3 nucleotides encode which amino acids? How many codons are there? This uses math we've already seen before. If a codon is 3 nucleotides, how many possible nucleotides are there at the first position?
4.
A's, G's, T's, and C's. So the first position can have 4 options. The second position can have 4 different nucleotides. The third can have 4. So that means there are 64 different combinations of 3 nucleotides if you're using a 4-letter alphabet. A's, T's, G's, and C's. Or A's, U's, G's, and C's. I apologize. So what are some interesting patterns that are important to note in the codon table? If you've seen this before, what do you know about the codon table?
There's more than one 3-letter sequence for the same whatever you call it.
Sure, there is redundancy. That is, if we have 20 amino acids and we have 64 codons, that means on average there's about 3 codons per amino acid, on average. What's the smallest number of codons you can have for an amino acid? That's the easy question. About 20 amino acids. What's the smallest number of codons you can have that encode an amino acid?
3.
Or you have to have at least 1 to encode it. So what is an example of an amino acid that's encoded by a single codon? Wait, what? I heard 2 at once, and it was an amino acid I wasn't familiar with. No, we already talked about one of them. Methionine. Only has a single codon. It has to be AUG. There's no other codon that encodes methionine. I think there's maybe 1 or 2 other ones up there. Don't remember offhand.
TRP.
Tryptophan. I'm a fan. You know why its single letter code is W? That's the things that are in. So we can talk about amino acids using their full names or a 3-letter abbreviation, which is what's shown in the cells, or a one-letter code, which is what's in the parenthesis. So do you know why tryptophan is W? Well, T was already taken. U is uracil, right? So we run out of with nucleotides and amino acids, we run out of letters pretty quickly. So somebody told me that the best way to remember that is it's tryptophan, which is not fair to people that have rhoticisms, which are people that can't say R's, but they say sort of a W-ish sound. So I'm not trying to make fun of anybody, but that's how I learned to remember that tryptophan is a W. Otherwise, yeah, T was already taken. What are the amino acids that have the most codons? The largest number of codons up here is 6. So serine has, I probably should have used a different color. There's 6 for serine, there's 6 for arginine. There may be another one that's, leucine. Leucine, I'm home. So there are a number, yeah, if you didn't get it, don't worry about it. It's an old joke, literally. So there's a range of 1 to 6 codons per amino acid. And then what's another important feature of the codon table? When you find codons that encode the same amino acid, what's similar about them, usually? Like let's pick on proline in red there. What's similar about its codons? They all start with CC. So the only difference is it doesn't matter what nucleotide is at the 3' end of that codon. It could be CCN. Any nucleotide encodes proline. And that's a general feature of the codon table if you start looking at those patterns, is most of the time when there are multiple codons that encode an amino acid, they're all in this same block here, which are, each of those 16 blocks are the ones that start with the same 2 nucleotides. So all the alanines are GC something. So you can have codons that have 1 codon per amino acid, 2, there's one that's got 3, which is weird, 4, and 6. You don't need to remember that. Last thing before we go, there are 3 codons up there that don't encode an amino acid. Those are the ones that say end up here. They're referred to as something else, also called stop codons. There are 3 stop codons. That is how the ribosome knows when to stop translation, is when it runs into one of those codons. And so we're going to pick that up more on Monday. And so before you go, let me tell you a little bit more about what to prep for Monday. Okay? I hope you'll still work on this. You may have tried it, you might not have gotten very far, you might have gotten to the end. The purpose here, and what I would suggest doing to work on this exercise, which is really important for the final if you decide to take it, and for your understanding of translation, is to this point we've looked at a lot of different DNA sequences that are important for translation, start and stop codons, transcription, promoter sequences, transcription start sites, polyadenylation signal sequences, splice sites. So the purpose of this exercise is to think about all of the DNA elements that have to be coded in the DNA that are necessary for transcription and then for translation to produce a gene that encodes those 7 amino acids, MPLEAS. So if you want structure for doing that, that's what I would do, is I would start with translation, know what do you need to put into a gene that would encode those amino acids, and then work back from there. What would you need to start and end translation of that sequence? What would you need to transcribe a sequence that contains those codons, and so forth. So for next time, we'll keep working on translation. There's a second video in the course manual. Please work on that. And one other exercise. Give it an attempt. And I will see you on Monday. Have a fantastic weekend.